This presentation aims to give a few insights into the evolving role of stem cells in the neurosciences. To understand stem cells, we have to get back to embryology. The fertilized zygote is totipotent. It produces the trophoblast in the placenta. The trophoblast is again pluripotent and can give rise to mesoderm, ectoderm, and endodermal cell lines. Then we have further evolution of cells into multipotent stem cells. These cells can proliferate along organ or tissue-specific lines. Some cells, which are highly differentiated, lose their proliferative potency. Neurons, for example, forgo their reproductive capability. This is known as Cajal's dogma. Let us see the embryology of the central nervous system. The CNS evolves from the neural tube. The neural tube is formed by an infolding of neuroectoderm, which is differentiated ectoderm. The brain is formed by a proliferation of cells at the cranial end of the neural tube. How does proliferation occur? The innermost layer of cells start multiplying. These cells proliferate. Then these cells migrate to the outer surface and then they integrate to form the cerebral cortex. The ventricles of the brain are actually invaginations of the neural tube, pulled along during brain differentiation. This helps us to understand that stem cell niches are found in certain locations, basically locations which are derived from the lining of the neural tube. You have subependymal zone of the ventricles. You have the dentate gyrus of the hippocampus, and even the ependyma lining the central canal of the spinal cord. If you want to use stem cells for repair of the nervous system, you can find them or use them from two sources. Endogenous stem cells are found in the dentate gyrus of the hippocampus. These stem cells are stimulated by enriched environments, by physical activity, and by certain growth factors. The subventricular zone and also the lining of the central canal also have resident stem cells with regenerative potential. Exogenous stem cells can be embryonic. These embryonic stem cells are obtained from aborted fetuses. They are pluripotent, but however, they have ethical issues. There is a problem of immune rejection, and teratoma formation may occur. Exogenous stem cells may be mesenchymal stem cells. These are derived from the bone marrow, from the amnion, from adipose tissue, or from cord blood. A newer addition to the field of stem cells is induced pluripotent cells. These are obtained from adult cells by genetic programming using retro or lentiviruses. Differentiated cells grown in culture can also be reprogrammed to pluripotent stem cell lines. Fibroblasts, astrocytes, and pericytes can be reprogrammed to neuronal lineage. How and where do we use stem cells? Stem cells can be administered by stereotactic injections to the site where they are required. They can also be given in the CNS by intraventricular injections or by intrathecal injections. In these situations, you want to use the local chemotactic factors which pull stem cells to areas of cell loss. So the clinical applications of stem cells include degenerative diseases like Parkinson's disease, inflammatory conditions like multiple sclerosis, amyotrophic lateral sclerosis. In each of these, the requirement of stem cells is different. In Parkinson's disease, you produce cells which are capable of producing dopamine. Neurotrauma also, there is a major role for stem cells. They have been used in trials in traumatic brain. So to summarize, embryologically, the nervous system is derived from neuroectoderm, which is differentiated from ectoderm. This neuroectoderm invaginates to produce a tube, which is called the neural tube. The inner layer, the neural tube, the lining layer, proliferates, and then it migrates to the outer surface. Rapid proliferation at the cranial end produces the brain. The ventricular system involves pouches of the neural tube being pulled into the evolving brain. So stem cell niches are found in the subependymal zones and also in the dentate gyrus of the hippocampus. Mature neurons do not replicate. This is Kajar's dogma. Endogenous stem cell proliferation occurs, followed by replication, migration, and integration. Exogenous stem cells can be embryonic or adult-derived. Mesenchymal stem cells can be derived from bone marrow or from blood. Induced pluripotent cells are derived from adult cells by retroviruses or by vector programming. The routes of administration of stem cells include intravenous, neurotrophic lateral stenosis. In neurotrauma, both traumatic brain injury and in spinal cord injury, and in cerebrovascular accidents, and even in cerebral palsy.